So, Alzheimer's disease is the leading cause of dementia, and unfortunately, there is no cure for the disease. So, if we can harness the power of autophagy, we would be able to prevent the disease or even mitigate this disease. Basically, we would be helping the brain heal itself. The study that we published shows that we can actually do that, at least in a mouse.
I'm Dr. Nathaniel Chin, and you're listening to Dementia Matters, a podcast about Alzheimer's disease. Dementia Matters is a production of the Wisconsin Alzheimer's Disease Research Center. Our goal is to educate listeners on the latest news in Alzheimer's disease research and caregiver strategies. Thanks for joining us. Welcome back to Dementia Matters. I'm here with Dr. Luigi Puglielli, a professor of medicine at the University of Wisconsin School of Medicine and Public Health, and a basic science researcher who studies age-related brain changes at the cellular level. He has published a series of papers on autophagy, what he describes as a natural garbage disposal within brain cells that removes bad proteins and sick organelles from the brain. When autophagy becomes disrupted, brain cells can clog with bad proteins, such as is the case with Alzheimer's disease. In 2009, Dr. Puglielli identified two enzymes that regulate the induction of autophagy from the endoplasmic reticulum, an organelle that makes a lot of proteins. Since then, he studied how these proteins work, and in 2016, he published a paper showing that the inhibition or blocking of these two enzymes can prevent Alzheimer's disease in the mouse. Last year, he published a paper further defining those enzymes and how they affect the autophagy process. More recently, Dr. Puglielli published a new paper that builds on his past discoveries and explains how restarting autophagy could lead to drug development for Alzheimer's disease. Luigi, welcome to Dementia Matters.
Thank you, Nate. It's a pleasure to be here. And thank you for the introduction.
You are welcome. I'm very excited to have you explain this very intricate process that you've really discovered. So, you have a new paper out in Communications Biology, one of the prestigious Nature journals. It's the culmination of more than a decade of research. So, before you tell us about the paper itself, can you share with our listeners how your career evolved into Alzheimer's disease and aging research?
Well, I've always been interested in aging and age-associated diseases. While in medical school, I trained in geriatrics, and as I moved from clinical research toward basic science, I remained interested in the general concepts of physiology and pathophysiology of aging. By studying the physiology of aging, we can learn why aging leads to increased disease vulnerability. While by studying the pathophysiology of aging, we can actually learn what happens in a disease that is highly associated with aging, such as Alzheimer's disease.
So, with that in mind, then tell us about your recent paper. And along the way, please explain for our audience any of those scientific terms that you're going to use, such as endoplasmic reticulum, enzyme, this ATACE 1 and 2, and then acetylation.
Okay, in general, the cell continuously makes new proteins to replace old ones that have been removed or degraded, or because they need to be secreted outside, for example, a hormone or digestive enzymes, or to simply perform normal activities that occur at the cell surface. For example, neurons that connect to each other to execute basic neuronal functions, which could be making of a new memory or recalling an old memory. So the endoplasmic reticulum is a very specialized organelle within the cell that makes a lot of proteins. These proteins are in general required to traffic through what is referred to as the secretory pathway, and this process and pathway is particularly important for neurons. Now, as in any process that requires building or assembly, some proteins will come out the right way, while others will come out the wrong way. The former, the proteins that came out the right way, need to be selected and shipped to their final destination, for example, to a synapse that is necessary to make a new memory or to recall an old memory. The latter, the proteins that came out the wrong way, need to be degraded or disposed of. If you let them accumulate within the cell, they will become toxic. Imagine not being able to remove your regular trash every day or week. It will simply accumulate, and at some point, living in the house will be unbearable. Now, to avoid these issues, the endoplasmic reticulum has a whole set of proteins whose job is to make sure: one, that all new proteins are made the right way; two, that only good proteins go to their final destination; and three, that the wrong proteins are disposed of. Basically, some sort of a quality control assembly line. Now, the biochemical machinery that we have identified and that we study is meant to do exactly that. It labels the good proteins with an acetyl group and then ensures the activation of what we call reticulophagy, which is literally autophagy from the endoplasmic reticulum. Now, autophagy or autophagy is a word that means self-eating. It indicates the ability of the cell to digest and recycle unwanted material, such as proteins that came out the wrong way and failed this quality control assembly line. So, to answer your question, ATS1 and ATS2 are two enzymes that acetylate, add an acetyl group to properly made proteins, allowing them to leave the endoplasmic reticulum and go to their final destination. At the same time, these two enzymes ensure balanced removal of toxic protein aggregates through the autophagy process. If this machinery works properly, all is good. If this machinery does not function properly, we run into trouble. So many chronic degenerative diseases that are typical of old age, including Alzheimer's disease, are characterized by the abnormal or excessive accumulation of toxic protein aggregates. So if we help our cells and tissues to degrade these aggregates, we would be able to resolve disease states. This is exactly what we show in our recent publication.
Okay, so that was a really complete answer. So thank you, Luigi. I'm going to ask some questions off of it. So we have neurons, and neurons need to move proteins in order to function properly. And this endoplasmic reticulum makes many of those proteins. Nicely, it has its own sort of quality control system that, there's efficiency. So if something isn't 100% efficient, you're going to have some bad proteins that are produced, but it can take care of those. It can kind of mark them with acetylation, and then it actually interestingly, it breaks it down itself and kind of gobbles it up and gets rid of it. And so that's sort of how a well-functioning neuron works. And when it doesn't work well, you have this buildup. And in the case of Alzheimer's disease, it would be this buildup of amyloid protein or tau protein, and that leads to brain cells not functioning well. So you've identified two really key enzymes or other proteins that are meant to actually help with the quality control and flagging these bad proteins per se that build up. Is that correct?
Yeah, correct.
Wow. Okay. And
You
so,
said it very well.
well, I feel like I'm learning a lot here of my biochemistry and my cellular biology here. But now you, in this most recent paper, you actually manipulated this process, right? So you were able to manipulate autophagy in the brains of mice and restart the process so that it could actually gobble up some of these bad proteins. And this opens the door, as you put in your paper, to potentially changing the timeline of Alzheimer's disease and other age-related neurodegenerative processes or diseases. So, can explain that part of it and why this discovery is really so important in the scientific community.
Yes, so Alzheimer's disease is the leading cause of dementia, and unfortunately, there is no cure for the disease. So, if we can harness the power of autophagy, we would be able to prevent the disease or even mitigate this disease. Basically, we would be helping the brain heal itself. The study that we published shows that we can actually do that, at least in a mouse.
And I love the idea of allowing the brain to heal itself. You are using the natural machinery of our brains to heal itself, to get rid of the toxic protein. But it's really hard to study Alzheimer's disease or at least translate findings from mice to humans. And we've seen that disconnect in the past. And so why is that? And is there a part of you that's concerned about the translation of what you're finding in mice to what potentially would happen in a human?
Yeah, well, first of all, you're correct. It's difficult to study this disease, human disease in mice. Mice do not naturally develop Alzheimer's disease. They simply don't make those specific protein aggregates that you mentioned, the beta amyloid or the neurofibrillary tangles, that are the underlying cause of the disease in humans. So, to circumvent this obstacle, we use transgenic mice. They were engineered to express the human proteins and therefore make the same or almost the same toxic aggregates that I mentioned. So it's a valid tool to study the pathophysiology of the disease, but also try to identify machinery or mechanism that can allow us to delay the disease or even cure the disease in mice before eventually going to humans.
And now you've already explained to us the ATACE proteins, enzymes 1 and 2. But are these in humans and do they increase as a person gets older?
Yes, so the ATS1, ATS2 are acetyltransferases, or simply put, they acetylate proteins. They simply move an acetyl group from the acetyl-CoA to a specific amino acid of the proteins. And as I mentioned, these enzymes are very important for cells that require very active secondary pathways such as neurons. So they're very important for the brain. We know that in the normal brain, they are regulated to adjust the secondary pathway in real time. So as we talk, I'm trying to understand you and sort of figure out what you're trying to tell me, and you are trying to do the same thing. And so we are adjusting the secondary pathway of our neurons in real time. We also know that these two enzymes are completely dysregulated in the brain of patients with Alzheimer's disease. So something goes wrong as a function of age and as a function of the disease. So they are simply higher and hyperactive in Alzheimer's disease brain, causing the accumulation of these toxic proteins. So if we can shut them down or at least go back to normal, we would allow the brain to reset this machinery and dispose of these toxic protein aggregates.
Well, I hope I'm not challenging your ATAs to be too high, Luigi. I hope I'm asking good questions of you. What I thought was really important in your paper too is that you talk about both of them, ATAs 1 and 2, but number 1, ATAs 1 seems to be a really pivotal one, and it can switch on in some cells. And I'm wondering how does that happen and what are things that might cause ATAs to switch on and decrease autophagy and lead to the accumulation of all these proteins?
We don't have the complete answer to that. We know that these two, so lower organisms have one ATAs. Higher organisms, mammals in general, have two. So we need both of them because we are a bit more complex. And so we have defined, we discovered mechanisms to regulate these two enzymes in a different fashion. So for example, we know that ATS1 can respond to the inputs of acetyl-CoA. More acetyl-CoA, more activity, less acetyl-CoA, less activity. ATS2 does not do that. So they've been designed to perform almost the similar functions, but also different functions. And so as we learn how to regulate these two enzymes, perhaps we'll be able to shut down only one enzyme and not the other one. For example, we know that ATS1 will be a better target for pathophysiology of age-associated disease. So if we can identify inhibitors, compounds that target only ATS1 but not ATS2, we can prevent disease and avoid any side effect.
And I think that's a key part because when I read your paper, you talk about the druggable properties of what you found, and it seems like ATACE1 is a druggable property, and that if we were to target that, potentially this could lead to clearance of amyloid protein or tau protein.
Correct, correct. And that's the one of the main conclusions of this paper is that we have a pipeline to actually select appropriate inhibitors and target them only to one of the two enzymes and move and study the mouse. So we have the perfect mouse because the mouse that gets sick in 2 months and dies in 4 months. And so right there, in only 2 or 3 months of work, we can actually test whether that compound is promising or not.
And I should clarify too, since I didn't ask this earlier, it seems like you actually can eliminate both amyloid and tau protein in mice. Is that right?
Yes, that is correct. Yeah.
So the two proteins that we define Alzheimer's disease, you've been able in a mouse to allow the brain itself to clear out those toxic proteins. That seems like such a pivotal and important finding.
Yes, it is, because there are compounds that can target one of these protein aggregates but not the others. And for the disease, they're both important, equally important, I would say. And so we need to be able to resolve both of them.
Well, then another question I have is there a negative consequence to doing something like this? So if you don't have Alzheimer's disease, let's say this is a normal mouse brain and you were to block these two enzymes, is something bad going to happen? I mean, is there a purpose to having ATACE 1 and 2 do what they're doing?
Oh, I'm sure there is a purpose to have both enzymes and not just one. In the mouse, we know that if you genetically shut down one of these two enzymes, the mouse is completely normal. So which means the mouse at least can live with only one of these enzymes. And there is no change in the lifespan. They're just protective from certain diseases. In humans, we know that there are humans that have a mutation in one of these two enzymes, and they are completely normal. The study that we have, these are mutations that inactivate the proteins, but these studies, these genomic studies, were done in individuals that are between the age of 50 and 80, which means they live basically most of their life without any disease manifestations. They are rare individuals. It would be nice to study them and to see what is actually happening, but it's sort of a proof that we can live with just one of them.
And what if we don't have either of them, though? Is there a problem if you lack both of them?
We don't have, well, I think it's going to be a problem. We know in lower organisms, if you shut it down, they die, so it's not viable, which means that we do need both enzymes. And as far as I can tell, there is no human being that has a block in both enzymes, so I assume that they're essential.
Oh, okay. And a very common supplement that people take is acetyl-CoA, and it's mentioned a lot in my memory clinic. And so I'm wondering, since you've talked about it, and in your paper you talk about acetyl-CoA, I mean, is there a similarity there, or what role does acetyl-CoA play in the brain itself?
Well, this is the acetyl-CoA that I talk about is the acetyl-CoA that is made expressly within the neuron, within the cell. So it's not the acetyl-CoA that is coming from the diet. Of course, the diet contributes to the pool of acetyl-CoA that we make. But this is fundamental for the brain, is important for metabolism, for energy, is the backbone for other metabolites, neurotransmitters in the brain. It's important for the acetylation of proteins in many compartments, not just the ER, but also, for example, in the cytosol in the nucleus. In the nucleus, for example, acetylation of certain proteins that are known as histones regulates transcription. So it decides what protein we make or do not make. So it has very complex functions that are extremely important.
Well, thank you for answering that because that's a very common question of why do some people take acetyl-CoA? And I know it's a supplement, so it's not regulated, and so I just wanted to get that answer out there. So thank you, Luigi. Now, how long have you and your lab been working, though, on this line of research, this idea of autophagy and endoplasmic reticulum and this whole process?
Uh, in short, since 2007, we came across the acetylation in the endoplasmic reticulum by accident in 2007. It was just an accident. And we immediately recognized the importance of this discovery. And so we decided to go after the machinery that acetylates the proteins because it was not known at that point, and we discovered the entire machinery between 2007 and 2010. And so after that we devoted our attention to the biochemistry, in other words, how the machinery works. We also made the necessary mouse models because there was none to study the biology. And the information that we collected suggested that these two enzymes, ATS1 and ATS2, were good targets for therapeutics. So in 2012, we ran a screen to identify inhibitors, and we came out with a paper where we described the first ATS inhibitors. In 2016, we were able to show that this inhibitor was effective in rescue Alzheimer's disease in the mouse. And then in 2018, we showed that the same inhibitors could delay aging in a mouse model of accelerated aging. So it went from 2012, when we identified the compound, to 2018 to know that it can actually be used in a model of aging. Now, last year we showed that if you genetically eliminate one of these two enzymes, the mice are protected from Alzheimer's disease. Now we describe a pipeline that would allow us to identify additional ATS inhibitors, and we also show that these compounds can revert, not just prevent, revert some age-associated diseases, such as Alzheimer's disease. You have to imagine that most of the patients will come at the clinic when they're having memory defects, so the process had already started, and so we need to be able to catch the process as early as possible, but also be able to revert and not just prevent it.
But wow, 2007. I mean, we're talking 15 years ago,
15
Luigi.
years of my life, yes.
One reason I wanted you to start with that answer is because I wanted to ask you the question of, well, why does it take so long to go from bench to bedside? I mean, why does it take, and many people ask this question of us, both in clinic and in the scientific field, and your answer, what you just said was part of it, but I would just like your thoughts, for those who ask, why have we not made as huge of steps? Well, one, I guess I would argue we have made huge steps, but two, I just want to know from you, what do you say to that? The slowness of the scientific progress.
Because there are many things that need to be done before being actually able to test a specific hypothesis. So as I mentioned a moment ago, we discovered the acetylation in 2007, but we didn't know the enzymes, so we have to identify the enzymes if we want to study them. Then we need to understand why we need acetylation in the endoplasmic reticulum. Why did nature decide to place acetylation in the ER? And then at that point, we did not have mice that we could use, so we had to make our own transgenic mice. and it takes time. So in other words, there was a lot to discover before reaching this point. And so since 2007, we published a series of 25 papers where we describe all this work. And now that we know in mice, we need to move forward into humans, but that is a very long line of work and is not easy.
Well, I hope you have time, Luigi, because it sounds like there's a lot to do here. But it does make a lot of sense. I mean, it's discovery, but then invention. You created your own cell line, and then more discovery and more explanation. But thank you for that answer, because I think that helps at least touch on the idea that in order to do the best science, it takes a very methodical, rigorous way of doing it.
Thank you.
So, what does the future look like for this line of research? You mentioned going into humans, but how do you see this unfolding and perhaps leading to drug discovery that might stall or prevent Alzheimer's disease?
Yes, well, I hope that the pipeline that we have described can help us identify more ATAs inhibitors. Unfortunately, 95 to 97% of all compounds that work in mice never reach the human stage. So 95 to 97%, that's a lot. So something happens across the line from mouse to humans that prevents them to be even tested. Therefore, if we wish to be successful and reach eventually the human stage, we need to identify many inhibitors, not just one, not just two or three. Furthermore, we definitely need to understand the specific details of the underlying biology so that we can envision disease states where we can employ these inhibitors, but also prevent possible side effects. And so there is still a lot to be done in this direction.
Okay, well then, Luigi, my last question for you is one that I like to ask all of my guests, and I haven't had a scientist or basic science researcher on before, so I'm excited to ask your opinion on this. Because you study a range of diseases of aging at that cellular level, what do you incorporate in your own life? What do you do in your personal life to keep your body and your brain healthy?
Well, first of all, this line of work keeps my brain always under what I call learning stress, under pressure. So I'm forced to learn new things every day. Forcing your brain to learn new things, example, a new language, is very helpful to preserve your memory, obviously. I also exercise. Outdoor aerobic exercise is excellent for your brain. If you think about it, our body and brain is designed to interact with the environment. As we walk, we run or bike, we must look at things, we must hear things, and we must adjust our walking, running, or biking to respond to the environment and the associated body stress, a bump on the road, etc. So, this is what the brain was initially designed to do: receive and process all this information that comes from the outside. Finally, I eat a lot of vegetables and fruit, but I do enjoy an occasional good meal.
Well, thank you, Luigi, for being on Dementia Matters, and we certainly hope to have you on again as your work progresses.
Thank you. It was a pleasure.
Thank you for listening to Dementia Matters. Follow us on Apple Podcasts, Spotify, Google Podcasts, or wherever you get your podcasts to be notified about upcoming episodes. You can also listen to our show by asking your smart speaker to play the Dementia Matters podcast. And please rate us on your favorite podcast app. It helps other people find our show and lets us know how we're doing. Dementia Matters is brought to you by the Wisconsin Alzheimer's Disease Research Center. The Wisconsin Alzheimer's Disease Research Center combines academic, clinical, and research expertise from the University of Wisconsin School of Medicine and Public Health and the Geriatric Research, Education, and Clinical Center of the William S. Middleton Memorial Veterans Hospital in Madison, Wisconsin. It receives funding from private, university, state, and national sources, including a grant from the National Institutes of Health for Alzheimer's Disease Centers. This episode of Dementia Matters was produced by Rebecca Wazaleski and edited by Kaylin Rauerdink. Our musical jingle is Cases to Rest by Blue Dot Sessions. To learn more about the Wisconsin Alzheimer's Disease Research Center and Dementia Matters, check out our website at adrc.wisc.edu. That's adrc.wisc.edu. Follow us on Facebook at Wisconsin Alzheimer's Disease Research Center and find us on Twitter at Wisconsin ADRC. If you have any questions or comments, email us at dementiamatters@medicine.wisc.edu. Thanks for listening.